So we left off with the end of transcription. We had our mRNA transcript, and now we will move on to translation, which is taking that mRNA message and producing the protein product. So as we saw with transcription, there's three stages: initiation, elongation, and termination. And remember, the ribosome is where translation is occurring. So this is either in the cytoplasm, or for a eukaryotic cell, it could be in the cytoplasm or on a ribosome that is embedded in the rough endoplasmic reticulum. So the ribosome is made up of ribosomal RNA and some protein. And there are two subunits that we call small and large because one is smaller than the other one. So we've already talked about this. This is not occurring in the nucleus, right? This is outside the nucleus in the cytoplasm or on the rough ER. Now, I mentioned the genetic code a little bit previously, and the thing that we need to understand is this is the conversion from a nucleotide language into an amino acid language. And the RNA alphabet has the four nucleotides: adenine, cytosine, guanine, and thymine. And for proteins that are made up of amino acids, there are a total of 20 different amino acids. When you take these four nucleotides and you look at how those could fit together in groups of three, because remember, we're talking about a triplet, right? Codon that's red. So for every three nucleotides, that codes for one amino acid. That's called a codon. There are 64 different codons, and most of those, the majority of those, are just coding for a specific amino acid, but there is one specific one, AUG, which is the start codon, and it actually codes for the amino acid methionine. And then there are also three stop codons. So these will be telling the ribosome, you're done, this protein is complete. So this is a genetic code chart so that you can see how does this translate from a triplet codon to an amino acid sequence. So let's say that let's just make up one here. A C A. So what we would do is we would come here. This is the mRNA sequence, and we want to know what amino acid that particular codon codes for. So we go first letter A, second letter C, so that's bringing us right into this box here, okay? And then the third letter C, oh, no, I'm sorry, third letter A, and there's our triplet codon. So we see that there's an abbreviation here, THR, and that stands for the amino acid threonine. So as you can see, let me just get some of this writing off of here. So as you can see, there are 64 codons there, and there are some overlapping. So in other words, if we look at this box right here, all four of those are going to code for the same amino acid. There's two more that actually still code for that same amino acid. So what we'll see toward the end is that sometimes that's a good thing, because if a mutation occurs, if the original sequence is CUU, and this particular nucleotide, there's a mutation and there's an error, and what we get is CUC, well, there's a mutation in the DNA or the RNA, but when that is actually produced into a protein, we won't see any effect because both of those code for the amino acid leucine. Now, the other thing I'll say is this genetic code is conserved for across organisms, so bacteria, plants, animals, same genetic code. So this is a cartoon representation of the ribosome, and what you can see is obviously the smaller subunit, the larger subunit, and then what we also see is how it comes together with, so this here represents the mRNA, okay, that was produced in transcription. This here represents the tRNA, the transfer RNA that's bringing in the appropriate amino acid. And then these look like very large beads on a string, Each of those represents an amino acid, so this is a growing amino acid chain. This is what is ultimately, this chain of amino acids, going to become the protein product. So, in the initiation stage, what's called an initiation complex is formed. And so we will see that the small ribosomal subunit will bind to the first tRNA. It's called the initiator tRNA. And I mentioned that there was a start codon, so the initiator tRNA is going to have an anticodon that is complementary to the codon of the start codon. So these, the small ribosome unit and the initiator tRNA, will bind to the mRNA strand, and then the large ribosomal subunit will join. Now, one thing that I want to point out, and we'll see the visual in just a minute, but there's essentially 3 sites on the ribosome where the tRNAs can come in and bring the amino acid. The A site, which stands for acceptor, that's basically the initial site that a tRNA is coming into the ribosome. The P site for peptide bond, this is where an amino acid that's brought in by a tRNA will be added to the growing chain of other amino acids. And then the E site, so once that tRNA has brought the appropriate amino acid in and it is shifted to the growing chain, then that tRNA will exit so that it can recharge with another amino acid ready to bring it back on the next round. So if we take a look here, you see that we have the mRNA sequence shown in orange, and this right here, AUG, if you remember, that's the start codon. This first tRNA that you see would be the initiator tRNA. And notice that it's complementary to the start codon. And the particular amino acid is methionine. Now, at this point, you see the small ribosomal subunit was there, and then the large ribosomal subunit will join, and at that point we move into the next phase, which is elongation. And for elongation, as the ribosome shifts along the mRNA sequence, the appropriate tRNA will bind. And so let's look at this. So we had those 3 sites that we identified. So this tRNA here is joining the A site. It's got an A on it. This tRNA is in the P site, and over here we have the E site. So nothing isn't quite in the E site yet. But as you see, once this amino acid joins here, then what we'll see is once this tRNA binds here, this amino acid, methionine, will actually there will be a bond that occurs between methionine and this other amino acid, phenylalanine, so that the amino acid is no longer bound to the tRNA. Instead, it's bound to the neighboring tRNA. And at that point, this tRNA is going to be shifted to the E site. You see here, it's shifted to the E site. It will exit the ribosome. Notice it no longer has methionine, but it can recharge and gain another methionine and come back and do the same thing. Now what you see is, so this amino acid, this tRNA, you see the codon for AAA right here, AAA, has shifted, okay, to the P site, and now you see it has 2 amino acids. Now the same thing will happen. These amino acids are going to form a bond. This phenylalanine will form a bond with this particular amino acid, arginine, and this will shift to the E site, and this one will shift over. So this is going to continue down the mRNA, reading them 1 codon at a time, and each time we'll have 1 more amino acid added to this growing chain. So elongation in words would say 1 codon at a time shifts. The tRNA initially comes into the A site, then it shifts to the P site. That peptide bond is formed. A peptide bond, recall, is just a bond between 2 amino acids. So that peptide bond forms so that those two amino acids are bonded together, and then that tRNA moves to the E site, is released, can find another amino acid. Now this is going to continue until the ribosome reaches a stop codon, which says it's time to stop. You have completed the protein, release the mRNA, release the protein. Once the mRNA is released, it can either be translated again, so in other words, it can find an initiator tRNA and a small ribosomal subunit and start the process again, or it may be degraded. mRNA is not intended to stay around for a long period of time. Remember, in cells, different proteins are needed at different times for different reasons, and therefore we want mRNA not to last a long time as conditions change. So let's practice. We have this genetic code chart. This is the sequence, I believe, that we had after our transcription activity. So we've already done this first codon, right? We know that that's methionine. That's the start codon. And we happen to have another one, okay, there. We don't have to do that one. But our next codon is UAU. So stop the video. Let me use the chart to find out what amino acid that is, and then start the video back. So hopefully you found that UAU is TYR, or tyrosine. And if we move along, GGC, you can stop the video again if you want. Make sure you can find that. But GGC is going to be here, and that's glycine.